this lesson, we will introduce two basic concepts in cellular communication and cell signaling. In multicellular organisms, cells possess a remarkable ability to communicate each other, coordinating their functions and responding effectively to their surroundings. This communication hinges on the utilization of signaling molecules known as ligands. These ligands, or primary messengers, are synthesized within cells and subsequently released or exposed to the extracellular environment, where they can be received by specific receptors on target cells. Once the ligand binds to its receptor, it conveys a message to the interior of the cell, triggering a response. Cell communication can be categorized into four main types: autocrine, paracrine, yuxtacrine, and endocrine. Autocrine communication involves cells sending signals to themselves. In paracrine communication, cells communicate over short distances with neighboring cells. Yuxtacrine or contact-dependent communication relies on direct or close physical contact between cells or with the extracellular matrix. Endocrine communication operates over long distances, with messengers traveling through the bloodstream, facilitating communication between different organs and tissues throughout the body. The messengers or ligands involved in cell communication exhibit specific chemical and physical properties. Hydrophilic ligands, such as proteins and peptides, are water-soluble molecules that can dissolve and interact within an aqueous environment. Typically, these ligands bind to receptors located on the cell surface. On the contrary, hydrophobic ligands, like steroid and lipid hormones, tend to avoid water contact. To navigate through the extracellular medium, they rely on carrier proteins. Upon reaching their target cell, due to their nonpolar nature, they can cross the plasma membrane and bind to receptors located inside the cell or intracellular receptors. Additionally, certain ligands, like gases, are very small molecules with the ability to diffuse directly across the cell membrane to also bind to intracellular receptors. The messengers or ligands released by cells don't activate randomly any receptor they encounter. Instead, there is typically a high degree of specificity in the ligand-receptor binding and activation process. When a ligand binds to its receptor, it triggers the activation of one or more signaling pathway cascades, also known as signaling transduction. These pathways comprise of a series of reactions that occur within the cell. Consequently, the cell will engage in a specific response. However, this response also depends on the cell type, environmental cues, and other stimuli. The cell is simultaneously sensing and integrating. During this process, various intracellular proteins and enzymes become activated or their function are affected. Additionally, small molecules, known as second messengers, can be produced to amplify the signaling cascade. These reactions ultimately impact effector proteins, directly involved in carrying out specific cellular processes. These can encompass the induction of gene expression, the regulation of metabolic pathways, and/or the alteration of the cytoskeleton, resulting in changes in cell shape and movement, among other potential processes. Cell signaling holds wide-ranging implications for the functioning of multicellular organisms. For instance, it influences essential aspects such as growth, development, physiological, and immune responses. Conversely, disruptions or malfunctioning in cell communication can give rise to pathological conditions and diseases, including diabetes, cancers, autoimmune diseases, among others. Now, let's examine some specific examples of the different types of cell communication. In the context of autocrine signaling, cells initiate a self-induced response by releasing ligands that binds to their own receptors. This mode of communication is observed in various biological processes, like cell division. An illustrative example can be found in T lymphocytes, which are cells from the adaptive immune system. These cells undergo a process termed T cell activation and clonal expansion. In this process, subgroups of T cells stimulate their proliferation by releasing cytokines, such as interleukin-2. These cytokines bind to the IL-2 receptor expressed on the surface of the same T cell. Once a specific number of proliferation cycles have occurred, the cell ceases to express the IL-2 receptor, thereby limiting their own division. Paracrine communication enables cells to coordinate responses within their local environment, with ligands acting as local mediators. Paracrine responses generally exhibit rapid but short-lived effects due to the rapid degradation of the ligands involved. An example of paracrine action is the vasodilator effect of nitric oxide. Within blood vessels, the inner lining comprises endothelial cells followed by a basal lamina and farther outward, smooth muscle cells. Endothelial cells can synthesize nitric oxide due to the presence of enzymes that convert the amino acid arginine into this gaseous compound. Upon release, nitric oxide can traverse the plasma membrane of the smooth muscle cells, binding to its intracellular receptor. This binding initiates a response leading to the relaxation of muscle cells, consequently causing the dilation of blood vessels. This physiological process has various implications, including the reduction of blood pressure. Though some authors categorize it as a type of paracrine communication, I would like to emphasize neuronal or synaptic communication as a distinct category due to its unique characteristics and significance. Nerve cells communicate through a combination of electrical and chemical mediators, including the release of neurotransmitters into the synaptic gap. Neurons comprise a cell body or soma, along with short extensions called dendrites and a long extension known as an axon. The signals between neurons are facilitated through electrical impulses that rapidly propagate along the neuron and continue in the subsequent cell through the release of ligands contained within small vesicles. These ligands include neurotransmitters that bind to receptors, such as neurotransmitter-gated channels, on the postsynaptic neuron, inducing the nerve impulse in the next cell. Yuxtacrine signaling occurs through direct or close contact between cells or with the extracellular matrix. For instance, cells can establish this type of communication via gap junctions, which are protein complexes formed between the membranes of two cells, allowing the passage of small molecules such as calcium ions. Another form of juxtacrine communication involves the complementary binding of protein complexes located on cell surfaces. The immune system frequently employs this type of communication in a process termed antigen presentation. For example, this can occur when a macrophage presents a fragment of a phagocytized or digested pathogen to a T lymphocyte. This presentation leads to the selection of lymphocytes for the previously mentioned clonal expansion process, among other functions. Lastly, endocrine signaling is a long-distance communication process wherein messenger molecules travel through the bloodstream to reach their target cells. Specialized in this mode of communication, endocrine organs like the pancreas, hypothalamus, pituitary and thyroid glands produce and secrete hormonal messengers crucial for the organism's physiological functions. For example, pancreatic beta cells release insulin, a peptide hormone serving as a hypoglycemic agent. Following a carbohydrate-rich meal, digestion converts carbohydrates into simple sugars. These sugars will be absorbed through the small intestine, consequently raising blood glucose levels. In response, the pancreas releases insulin, which travels through the bloodstream to reach receptors on target cells, such as skeletal muscle cells. This prompts the mobilization of glucose transporters to the cell surface, enabling cells to capture glucose for subsequent metabolism, and reducing available glucose levels in the blood. In this lesson, we've delved into various aspects of cellular communication and cell signaling. We explored the different forms of cellular communication, including autocrine and paracrine communication, the distinctiveness of neuronal communication, juxtacrine and endocrine communication. We highlighted that cellular communication is facilitated through the interaction between primary messengers or ligands and their specific receptors. This interaction leads to the activation of cell signaling pathways, characterized by the reception of the message, the activation or modification of signaling proteins, the synthesis of second messengers, and the activation of effector proteins. Ultimately, these processes culminate in the engagement of a particular cellular response. If you have found this lesson useful, please consider supporting this channel by liking the video, sharing it, subscribing to the channel, and leaving a comment.